Hello everyone, in this video I'll be explaining on the topic of smear preparation, classification of staining and simple staining. Coming first to the smear preparation, here it can be prepared from two sources, either the liquid media or the solid media. If it is from the liquid media, that is from broad culture, using a cooled sterile loop, place a loop of the broad containing the bacterial cell into slide that is of grease-free microscope and spread in a circular motion to about 1 centimeter in diameter. Here it can be done using a pasture pipette, a clean pasture pipette. Take a few volume of the bile from the broad culture inside the test tube and place 1 drop of it on a clean glass slide and spread it and make a smear and then allow it to cool down at room temperature. Next is it can be prepared from the liquid media or it can be prepared from the culture plate. Here, place a drop of saline water on a clean glass slide. Select the isolated colony to be stained and using a sterilized inoculating loop. Transfer an isolated colony from a culture plate to a slide. Be careful to touch only the top and the center of the isolated colony gram stained, and then use the loop to create an emulsion. Disperse the bacteria on the loop in the drop of water on the slide and spread the drop over an area the size of a dime. It should be a thin, even smear, allowing the smear to air dry thoroughly. Then heat fix the smear cautiously by passing the underside of the slide through the Bunsen burner flame 2 or 3 times. Heat kills and fixes the bacteria on glass slide due to coagulation of bacterial proteins. It fixes the cell in the slide. Do not overheat the side as it will distort the bacterial cell. The fixed smear is stained by appropriate staining technique. Next, coming to the common staining techniques. Here, first is the simple stain, which can be used as a quick and easy way to determine cells, shape, size, and arrangement of bacteria. This, true to its name, the simple stain is a very simple staining procedure involving high solutions and stains. Basic dyes such as methylene blue or basic fuchsin such as methylene blue or basic fuchsins can be used as simple stain. They produce color contrast but impart the same color to all bacteria in a smear. Next is negative staining. Here the bacteria are mixed with dyes such as India ink or necrocene. The background gets stained and unstained bacteria stands out in contrast. This is useful in the demonstration of bacterial capsule which do not take simple stain. Impregnation method. Bacterial cells and structures that are too thin to be seen under the light microscope are taken by impregnation of silver on the surface to make them visible. Demonstration of bacterial flagella and spirochetes. Next is differential stain. They impart different colors to different bacteria or bacterial structures. The two most commonly applied differential stain are the gram stain and acid fast stain. Coming to the simple staining. Here, the simple stain can be used as a quick and easy way to determine cell shape, size, and arrangements of bacteria. True to its name, the simple stain is a very simple staining procedure involving single solution of stain. Any basic dye such as methylene blue, safranin or crystal violet can be used to color the bacterial cells. These stains will readily give up a hydroxide ion or accept a hydrogen ion, which leaves the stain positively charged. Since the surface of most bacterial cells and cytoplasm is negatively charged, these positively charged stains adhere readily to the cell surface after staining. Bacterial cell morphology, such as shape and arrangement, can be appreciated. Coming to the principle of simple staining here. In the simple staining, the bacterial smear is stained with a single reagent which produces a distinctive contrast between the organism and its background. Basic stains with a positively charged chromosome are preferred because bacterial nucleic acid and certain cell wall components carry a negative charge that strongly attracts and binds to the cationic chromosome. The purpose of simple staining is to elucidate the morphology and arrangement of bacterial cells. The most commonly used basic stains are methylene blue, crystal violet, and carbolfuchsin. The reagents and equipments that will be required for this simple staining are methylene blue, crystal violet, and carbolfuchsin, which stains are to be used, and then spirit lamp or Bunsen burner, inoculating loop, staining tray or staining rack, microscope and glass slides. Coming to the procedure, place a slide on the staining tray or rack and flood the smear with one of the indicated stain. Using the appropriate exposure time for each carbolfuchsin 15 to 30 seconds, crystal violet 20 to 60 seconds, and methylene blue 1 to 2 minutes. Gently wash the smear with tap water. In this way, you can reduce the loss of organisms from the preparation. Air dry the slide and observe under the microscope. Coming to the result interpretation, here the bacilli and diplococci will appear rod shape when purple, spirula will appear spiral shape, bacteria purple in color, cocci will be spherical shape, bacteria purple in color. So this is the result interpretation of simple staining. So the color will depend on what stain you use for this simple stain. In simple stain, only one stain is used where all the structures get stained by that particular stain. So here if you are using the crystal violet, all the organism will appear violet in color, violet or purple in color. Okay.